And now it's really my honor to introduce our today's keynote speaker, Carl June. Carl has won many, many awards. Among them, he's been recognized by Time Magazine as being one of the most 100 most influential people alive. So we are lucky to have him. I think even more striking was who wrote the letter for Carl when he was nominated by Time as one of the 100 most influential people, who was Emily Whitehead, who was a young woman whose life was saved by Carl by a CAR T-cell therapy that he helped to develop. And just speaking personally, I have to say that I am inspired by Carl, and again, I've watched his relentless efforts to use every piece of information about what we know about the human immune system and new technologies, synthetic biology, to try to develop cellular immunotherapies for cancer. And so Carl is not only an inspiration for me, but really for the whole field of genomic immunology. And it seems absolutely appropriate for him to be the keynote speaker today for our institute. So thank you so much, Carl, for joining us. And let me hand this over to you.
Well, thanks, Alex, for the introduction. And I really want to say, congratulations on the wonderful, new Institute of Genomics and Cancer Immunotherapy. I think it's just, it's a great testimony to your leadership, and, and you have, just by the agenda, you put together an amazing set of studies and investigators that are going to lead lots of innovation. And disclosure here, I'll be discussing Novartis data. Next slide. And this is an outline of what I'd like to go over during this, and hopefully have questions and discussion at the end. Next slide. And the CAR field didn't just happen overnight. There's been about 30 years of research with some of the prominent people listed here that I've been privileged to know and who have and mentored me in some cases, and other cases are great friends. And it's now a large commercial industry, lots of biotech, and but these are all white men. Next slide. What a lot of people forget is that early on there were some women involved in this as well, and many now in faculty, both at my institute and yours as well. And they were all involved in the initial biotech with CAR T-cell development, and then biotech exited that in 1999 when there was toxicity in gene therapy trials, and no one thought that gene therapy could actually become a commercial product. And so those companies all terminated their research, even though they were leading the field at that time. So now we're at a really a very different time, but that's for historical reasons. Bring this up. Next slide. So at this audience is very sophisticated and doesn't need to. But I'm going to today focus, with one exception, on CAR T cells. But I believe, in fact, both are going to be used routinely in cancer immuno-oncology, both, MHC restricted transgenic TCRs, and as well as CAR molecules. And in fact, they may be used orthogonally, I think, because they have different mechanisms of resistance for emerging data. Next slide. And, so as I meant, 30 years ago, CARs were beginning to be developed, initially actually in HIV. And then both my group and Steve Rosenberg's group, showed first clinical data of real clinical activity back in 2010 and 11, and then that led to FDA approval. And then this year now, not moving beyond CD19 for FDA approvals for myeloma, target, and BCMA. So, and I think we're going to see a large, continued pipeline of approvals. Next. The initial immuno-oncology was heralded back in 2013, and Michelle Sadelain and I wrote a review back a few years ago now of showing the very high response rates to various B cell malignancies with CARs made at different institutions. And so there's now a large effort on this, and the initial results that we had been confirmed now and extended into many different disease settings. Next. So today I will discuss the process of autologous CAR T cell therapy and TCR therapy, which is cumbersome but can yield products that are very potent and has durable remissions. But I think we're going to have both off-the-shelf cells as well, derived from cord blood and healthy donors and iPSCs. And I actually think they'll have different indications and be with different advantages. We have just opened a trial here at Penn with a triple edited allogeneic CD19 CAR T cell using standard CRISPR-Cas9 gene editing technology. And there's many centers now that have different forms of either NK cells or T cells that are allogeneic and can be off the shelf. So I think it'll be a broad intent as we go forward. Next slide. And, so this whole field began, I think, from my own biased view as a bone marrow transplanter, from the realization that we had a graft versus leukemia effect, and that contributed to long-term remissions in AML in particular. But the field also came from the field of TIL cells with, and now we recognize their repertoire and could induce remissions in some patients with metastatic melanoma. And so many different natural cells have shown safety and some efficacy. And where we're headed, I think, is what the meeting theme is about, which is genetic editing, gene transfer, synthetic biology to make all kinds of cells that'll have both enhanced potency as well as improved safety. Next slide. So our initial patient that was infused back was July of 2010, led to a series of publications in adult and acute and chronic lymphocytic leukemia. And then in 2012, we treated, as Alex mentioned, Emily Whitehead, the first pediatric patient. And we had actually developed the CAR for ALL. That was the samples we used, but we weren't allowed by the FDA to treat pediatric patients until we had safety data in adults, which is why it was delayed. And that led to the FDA approval in 2017. And the reason we made a CAR with a 4-1BB co-stimulatory domain was that Marcella Maus was a graduate student in my lab back then in the early 2000s, and we studied the functional activities of 4-1BB agonism and compared that to CD28. Next. And just click. Is there a click? Anyway, what Marcella showed, and the clicks aren't showing up on my, is that we could get long-term proliferation of human CD8 cells for months using 4-1BB agonism, and it was actually polyclonal CD8 cells. And that's why we started with the initial trials. The two commercial products for CD19 are compared and contrasted here. Lentiviral vector versus retroviral engineering. The binders are the same. Promoters are obviously different, and then the signaling domain's different. And there's a very major difference then in how long these CAR persist in patients. And I think actually we want to have cells that have either short or long-term persistence, depending on the clinical indication. And one way to do that is just to make a cocktail of cells that have different signaling domains. There's much, I think, of research here to fine-tune this. Next. So, and then in 2012, after our initial CLL patients treated in adults, we treated Emily Whitehead and had a shock actually, which is shown on the left, where she had a cytokine storm beginning 3 days after we treated her with very high levels of interleukin-6 and interferon gamma. Quite surprisingly, TNF alpha was really only a minor player in this syndrome, and that led to our first use of tocilizumab and an IL-6 receptor antagonism, which was co-labeled then to treat the cytokine storm, and we're now using that preemptively here at Penn and Children's Hospital. Emily's now a junior in high school and a straight A student with an unmaintained remission in her leukemia. She's been vaccinated for COVID with the FDA-approved vaccines and likely has not responded, as I'll get to, because she has B-cell aplasia and likely cannot, as long as she has B-cell aplasia, be vaccinated against novel, B-cell to make antibodies where she doesn't have a plasma cell memory pool that would be CD19 negative. Next slide. And the first adult patient back in is shown here when we infused him at the end of July, Bill Ludwig, who had a P53 mutant and chemotherapy refractory form of CLL. And I don't know if you can do it in advance. Yeah, so that, thanks. So he came back last August in the midst of the pandemic and still leukemia free. And the reason he's sitting in an infusion chair is every 3 months he was getting gamma globulin. Next. But then 3 months after that visit, he got from a contractor in his house COVID, and he passed away this February from COVID. We now have a trial here at Penn led by Vijay Boj, studying the response of cancer patients and CAR in particular patients, their responses in great depth after vaccination with various vaccines. Next slide. And with Joost Mellenhorst, we've had, and Joe Faretta, a number of studies on these long-term patients treated, in the early 2010s. And Bill Ludwig had persistence of the cells back in August of 2020 that we could detect with this anti-idiotype. And he had a dominance at that point of CD4 CARs, but some CD8 CARs in the blood. We don't know really in the bone marrow. He was in remission. But for the first 2 years, he had dominant CD8 CAR T cell persistence, and then gradually the CD4 cells became the predominant cells, at least in the blood, expressing the CAR. Next. And so this is still unpublished work with a follow-up of that patient. We have done now TCR site-seq analysis on the circulating cells in Bill Ludwig. Click next. And very surprisingly, I think, we find, he has endogenous cells circulating that are not CAR modified, and then he has his, 1 or 2% of the cells that are still the persistent CD19 CARs. And what you can see from these UMAP projections is that these cells, first of all, they're polyclonal as far as their TCRs, but unlike the endogenous T cells, they're in S phase and G2 and M. So very different than how other memory and persisting T cells, where they have intermitotic long-term survival. The CAR T cells in these patients long-term, at least with CD19 as a target, are maintained by a slow tick over proliferation and click. So that's, to summarize, these cells are proliferative. They come to a steady state. They have balanced checkpoint molecule expression, for instance, TOX, PD-1, TIGIT, CTLA-4, but they retain anti-tumor activity, meaning they're CD19 specific killer cells, and they have features of memory cells such as active mitochondrial fatty acid oxidation. Next. And so, in summary, the long-term survivors and adults have, many have had durable remissions without other bridging or maintenance therapies. The field has found that you can maintain or pick survival of the CAR cells depending on the signaling domains. And so we think in long term, at least with CD19 as a model, that probably there's pre-B cells in the bone marrow that drive CD19 dependent proliferation of these cells, and that we can't find any targets that are CD19 positive malignancies, of the original tumor. This is probably very different than, CD19 is probably a special case, I think, with what we're seeing in myeloma and other solid tumors is not this, where if you would get a remission and all the antigen would go away if it were not on a healthy cell that can act like an antigen presenting cell, probably it's a very different equation here. So that we have, I think that's a major actually issue for the field to solve. Next. So, we found in Emily Whitehead this very prominent multi-organ failure that was rescued with tocilizumab. And then cytokine storm became a major issue beyond just CAR T cell patients in the field of medicine in general, with COVID and the realization that some of the patients in the ICU are having cytokine release syndromes. And it's a very complex issue that I reviewed in the New England Journal last year, of many different cytokine pathways that can be triggered by different issues such as uncontrolled viral infections or various HLH like congenital syndromes, and then lead to effector cytokines that cause the tissue damage. But one thing that's been unexpected with a cytokine release syndrome with CD19 CARs has been the nervous system symptoms, which usually happen after the general cytokine release syndrome. Next. And this is the actual package insert and black box warning for Kymriah. And it has, the three unusual things. It has a cytokine release syndrome, which is also seen with BCMA CARs. And we've seen it in solid tumors as well, and we can discuss that. And then this unexpected neurologic syndrome. And something a lot of people don't realize is that you can have a false positive HIV assay in patients who have engraftment with the lentiviral modified CAR cells because of the backbone sequences and the lentivirus used to make the CAR molecule. Next. And, in work that my lab did with Ansu Sapathi, who will speak later, I think, and Howard Chang's lab at Stanford, we had a very unexpected finding that this so-called ICANS syndrome, or immune effector cell associated neurotoxicity syndrome, may well be related in part to the unexpected expression of CD19 in the brain. And so all the textbooks would say it's a PAX5, driven expression in B lineage cells. But what Howard and Ansu found is with single cell mapping that the brain mural cells, the pericytes, actually express CD19. And in work that we still haven't published that Avery Posey's led here at Penn, you can find in syngeneic mouse models that this can lead to blood-brain barrier leakiness, and probably contributes then to some of the neurotoxicity in patients, which is fortunately reversible in almost all cases. So what's really also unexpected here is this expression in parasites is limited to the CNS. If you look at the parasites in your lung or in their, for instance, the veins in your arm, they don't have CD19 expressed. So there's likely some unknown function of CD19 in the brain, maybe a neuroimmune form of communication. Next. So there's a lot theme in today's meeting about genome editing and synthetic biology. One of the few instances where a new form of cell therapy was done outside of cancer was in the case of HIV, AIDS, actually, where about almost 10 years ago, we used with Sangamo zinc finger nucleases to knock out CCR5 in the CD4 cells of patients and showed that that was safe, so that a single gene could be knocked out by NHEJ and lead to the persistence long-term in patients with HIV/AIDS of cells that are resistant to infection with the CCR5 tropic HIV virus. But there are many things that are being talked about at this meeting where we could enhance the function of T cells with knockouts, as well as gene deletions, and as well as, making universal CAR cells. Next. And so we, and a large group of people, including some at Stanford, have, did as a proof of concept with CRISPR-Cas9 a trial using 3 guide RNAs with a T cell receptor that was HLA-A2 restricted to the cancer testis antigen YORK-ISO1. Next. And in this trial, shown on the upper left, were, we intended to do 4 gene modifications at once to the autologous T cells. So with guides against the T cell to track TRBC and then PD1 or PD-CD1 gene, as well as the lentiviral knock-in of the YORK-ISO1 TCR, which we showed worked better if the endogenous genes were knocked out. And we found in 4 patients enrolled on the trial that we could get good expansion of these cells. And this is with the first generation CRISPR-Cas9 that did out in the lab and made back. And, we were used what we had available back in 2017, for this manufacturing. We can now do this at much higher levels of efficiency with the current nucleases. But on the lower left really was the thing that was we'd hoped, where with single cell sequencing of 27 cells here on a modified 10x genomic platform that Ansu and Howard Chang made, we could not only see if there was the gene addition from the lentiviral vector to express the YORK-ISO1 transgene, but whether or not at the single cell level, the different genes were knocked out. And 6 out of these 27 had all 3 knockouts and other, you can see the Venn diagram of the frequency of digenic and monogenic editing. So in this trial, we found at the level of a single cell that multiplex editing is possible with very detailed analysis that Rick Bushman's lab did here at Penn. We found that off-target edits were very rare, but we did find chromosomal translocations up to 2% of the cells that were infused had translocations, but all 3 guide targets here that track the TRBC and PD1 were all on different chromosomes, so that it's not unexpected on the repair process. What we did also find was that those cells that had translocations had decreased fitness, and they decreased in the patients over time compared to the cells that were gene edited but did not have translocations. And then we are now working, and I'm sure many of you at this meeting, using base editing technology where to do the same synthetic enhancements, but not with avoiding double-stranded breaks. Next. And probably the main impact for the field behind showing that it's feasible to do multiplex editing is the survival data of the 3 patients who were infused. 1 patient died before they could be infused, and 3 patients were infused of the 4 enrolled on the protocol. And there we had long-term persistence of these TCR engineered cells, which really showed that the use of the bacterially derived Cas9 did not render these cells significantly immunogenic. And we found also very good trafficking. 2 patients, 35 on the left and 07 on the right, had myeloma, and bone marrow biopsies showed that the TCR modified cells had trafficked into the bone marrow, and then patient 39 had a synovial cell sarcoma, and that patient had a single biopsy which showed the presence of these gene modified cells in the tumor, similar to the frequency that was discovered in the peripheral blood. So that's I think bodes well for other forms of genetic modification of infused cells. Next slide. And so in summary, it's feasible to do this with both gene additions and gene knockouts. And off-target edits are rare and look very much as similar as what we've seen in others with small-scale experiments at the bench. Translocations in T cells, at least, they have decreased fitness. We've never been able to transform peripheral blood human T cells with a variety of genetic manipulations. And I think base editing technology now is going to be a great addition to this approach. Next slide. And so what about ways to further enhance the ability of CAR T cells besides gene editing? Next. And there were the 10th patient we ever treated was a 78-year-old man with CLL. And he had an initial infusion of CAR T cells, which did not persist well. And we remanufactured his product and then gave a second infusion, at which point 50 days after that infusion 2, as shown in the middle panel here in the upper left, he had cytokine storm that was treated with tocilizumab. And then he was discharged and then had a complete remission of his abdominal and masses shown on the CT scans, as well as disappearance of leukemia cells from the blood, as shown in the upper right there. Click. And, so Joe Ferreira investigated this in detail with at that time as a postdoc in my lab, and now he's on the faculty here at Penn. And we found that the CAR had integrated authentically into the TET2 locus, leading to loss of function mutation of that gene. Click. And we showed that, in fact, there was a clonal outgrowth in this patient where initially he had, for instance, if you looked at the whole blood, many different integrants containing the CAR transgene across all the TCR V beta family members. But if you looked 2 months after the second infusion at the CD8 cells, there was only one TCR, and it was a TCR V beta 5.1 cell. He was an HLA-A2 homozygous patient, and we were never able to determine what this T cell receptor recognizes. We think it's probably a passenger here, based on our subsequent experiments that show that loss of function of the TET2 was probably more relevant to the clonal outgrowth. Next slide. And so Joe Ferreira with Matt Porteous's lab at Stanford now has been working on intentionally doing this, and he's using AAV6 and a dual knock-in strategy to knock in a CAR to the track locus, and in this case, EGFR reporter gene into TET2. Next. And this strategy can be quite efficient, as shown on the left here, knocking in the Kymriah CAR into track, or knocking in the EGFR reporter into the TET2 locus. And then when you take these cells and stimulate them with leukemia targets that are CD19 positive, this shows in the 5 repetitive stimulations over 3 or 4 weeks. Beginning on the 3rd stimulation, there's a difference between the cells that have the single knock-in, as Justin Equim will discuss, I think, later today, or into track, or if they have a knock-in also into TET2. There's restrained differentiation in the cells that also have the TET2 knock-in, and as shown by CCR7 and RO plots here. So that phenocopies what we found in the patient. And then Joe's lab has shown that here with cells that have this double knock-in at about a 30% efficiency, that over time the CD8 CAR cells begin to dominate in the culture, whereas initially in this particular experiment, they were CD4 cells. And they proliferate better, as shown in the bottom center, if they have the dual knock-in. And it's not an immediate effect, it's something that comes out later, which is what we showed and found in the patient with the accidental disruption. So they have a growth advantage in the CD8 CAR cells appear to phenocopy in what we've seen so far in the patient with the accidental disruption. Next slide. And so Joe has tested this further here after restimulation and shown that the cells are very potent killers of CD19 expressing leukemia cells across this E to T ratio, and that they also make a lot of effector cytokines. So they look like central memory cells, but are capable of secreting interferon gamma and TNF alpha. Next. So in summary, from this patient 10 infused back in 2014, we found the clonal outgrowth of a single cell. If you look at manufacturing, that could say potentially that this whole field of cell manufacturing could be perhaps made much more economically affordable with the cost of goods if the single or a few cells could be used, although it would take longer for the cells to grow out in the patient. We found that this unintentional knockout was responsible for CAR T function and can phenocopy this with knock-ins. And I think perhaps most importantly is these CAR T cells and TCR T cells, since they're genetically modified, enable a very potent way to really screen the human genome with reverse genetics. So it's very important that all centers that are having trials with CAR T cells maintain banks and look for outliers like this, both for cells that have enhanced function as well as dropouts that may be the undesirable sites. Next slide. So the real issue facing the field, though, is I think all heme malignancies will respond to genetically modified TCR and CAR T cells. The issue is really safety and preventing antigen escape. But with solid tumors, there are many other issues that are required to be solved. Next slide. And in an attempt to do this, my lab's been working for several years now with an in vitro model where we use pancreatic cancer cells and then a CAR targeting mesothelin and using repetitive stimulation in vitro to identify in the Venn diagrams how much of CAR T cell dysfunction is related to what's been well described for chronic viral infections, for TILs and cancer, and for what might be specific to CAR T cells alone. Next. And so in work, this is from a postdoc in my lab, Shun Kuromitsu, who's now returned back to Japan. But what he's done is repetitive stimulation with a KRAS mutated pancreatic cancer line, ASPC1. And then those cells are called chronic antigen exposed, or CAE, and then studied these and compared their genetics and epigenetics to the input cells and to cells that have rested and not been continuously exposed to antigen, as may happen in patients. Next slide. And when he does this on the upper left with 5 normal human donors, the CAR T cells stop dividing after about 3 weeks, even though they continue to express the CAR. And this is unlike, for instance, if we would use CD19 CAR targeting leukemia cells, where the CAR T cells will continue to divide. About half the CAR T cells on the upper right lose surface expression of the CAR, although they continue to have the transgene and the RNA for the CAR. They, on the lower left, stopped. They lose the ability to kill target. And the green line shows the initial cells, where if pancreatic cancer cells by excelligence, all the tumor cells are eradicated within 2 days, but the cells after 3 weeks of the in vitro exposure to CAR T cells can no longer kill compared to, say, an irrelevant CD19 CAR. And they, as shown on the lower right, the cells stop producing cytokines when they see targets. And that's similar to many viral infections with dysfunction. Next. And when we look by single cell and bulk RNA-seq, there's about 1,000 differentially expressed genes. And quite surprisingly in Ingenuity pathway analysis, 3 of the 5 top pathways were actually came up for innate immunity. That was genes were induced for in natural killer-like cells, as well as some of the things we expected with T cell exhaustion signatures and, for instance, PD-1 pathway genes. Next. And on the upper left, what we did was carefully curate 4 papers and that had been where TILs had been analyzed. So patients given in the natural course of their cancer, the TILs were analyzed from lung cancer, colorectal, melanoma, and liver cancer. And many different genes were found to be differentially expressed, and 18 were in common between all 4 of these studies. And on the upper right, those 18 genes are listed. And we looked then on this heat map at day 28, chronically antigen exposed CAR T cells and with this pancreatic cancer target compared to the input cells. And you can see many of the genes are upregulated that are commonly upregulated in patients with various solid cancer. But most surprisingly though is this induction of NK like genes, which is shown in panel D at the bottom. The resting day 0 product, these are CD8 alpha beta T cells, and after 28 days of exposure, many NK genes are expressed. Next. And this is work with Louis Lanier's lab at UCSF, where with his CyTOF and curated NK panel, we find that the day 29 chronic antigen exposure cells are very different as far as their NK molecule and exhaustion molecule expression. And this just shows at 2 time points. We're also then looked at human samples. So on the top right is actually from patients treated in a New England Journal patient that had a paper with the CD19 CAR. These patients were given infusions. They had refractory lymphomas. And you can see patient 39 on the left, at a month after infusion, almost 30% of his circulating CAR T cells were CD56 positive. And a number of genes that are, NK markers are shown on the bottom for patient 39 after 27 days that weren't expressed on his manufactured product. The kinetics of the induction of these different NK-like genes varies depending on the gene that's being looked at. Next. And when we do UMAP projections with single cell sequencing of, in this case, 20 days after exposure, 4 clusters emerge. 2 of the clusters are what you would see and anticipate with more similar for exhausted cells. That's clusters 2 and 3 in the middle panel. But 2 other clusters are these dysfunctional NK-like cells where they express a number of NK markers and they're dysfunctional. And surprisingly, they express the transcription factors ID3 and SOX4, which are not really a lot known about their function in human CD8 alpha beta T cells. And we did with Shelley Berger's lab here at Penn epigenetics with ATAC-seq. There's on the resting day 0 CAR T cells, that gene's not expressed ID3, but there's a lot of reads back on day 28. And similarly, by epigenetics, there's a lot opening up of the ID3 locus after 28 days of stimulation with the antigen that where the locus is closed on the resting functional CAR T cells. Next. So in summary, this work I think is a relative, at least for pancreatic cancer, useful model for the dysfunction that occurs with CAR T cells, in this case, mesothelin-specific CAR T cells. We identified this upregulation of ID3 and ID4, and if we knock those out in T cells, it makes them partially resistant to this dysfunction, and they have prolonged killing and proliferation. And we've, as I mentioned, found this signature in human and patients treated both on our pancreatic cancer trial as well as the lymphoma trial with the data that I showed. Next slide. This is work from Lex Johnson, who was a graduate student with my lab and Andy Nins, and then a postdoc in my lab, where he was working with in a mouse system. And Andy had discovered that the gene 7SL, which is an abundant 7S RNA and a highly structured RNA, when it's expressed in T cells, it activates innate signaling and in dendritic cells as well. And so what Lex did was to follow up on the initial work done in Andy's lab, where he found that 7SL activated innate signaling, was to express 7SL in CAR T cells, and found that that could be exported in extracellular vesicles and recruit in dendritic cells into the tumor microenvironment. Next. And so 7SL it's part of the signal recognition particle. It's a very ancient RNA, and in tumors overexpress it, but it's unshielded in tumors where next click, where normally in health, the 7SL RNA transcript is the RNA is bound by many different proteins, which in one function prevents innate recognition by MAVs and RIG-I. But in cancer, the transcript is overexpressed, becomes unshielded by the proteins, and then activates signaling, and particularly through MYC signaling pathway in cancer cells, and in T cells and tumors, dendritic cells, it activates immune stimulated gene responses. Next slide. And so in a paper published a few months ago, Lex shows that this RNA can be packaged with a CAR, in this case against B16 tumors, and that the packaged RNA is exported in endosomal extracellular vesicles and taken up, where it then gets cross-priming and is able then to provoke the rejection of tumors that are mixed, where they either have the CAR target or don't. And so this is something we're scaling up for a trial next year here at Penn. Next slide. CAR T cells actually started in HIV and AIDS. And Jim Riley, whose lab is next to mine here at Penn, had a paper taking further the CCR5 edited cells and showing how it can enable the control of HIV replication. There are a number of companies now that have CAR T cells and other forms of engineered cells for autoimmunity and organ transplantation. One of my graduate students, Michael Klaczynski, made what was Sargil's lab, CAR macrophages, where he's expressing a CAR and shows that these macrophages then can phagocytose tumors in an antigen-specific fashion. And that's now in a trial here at Penn using HER2 as a CAR. And I think that will be orthogonal and with T cells, because of the very different killing mechanisms and potential for antigen presentation, that combination of both NK cells and macrophages may well be appealing. And I'll just finish with some ongoing work here on the use of CAR T cells for fibrosing diseases. Next. So this is work that we began with John Epstein's lab here at Penn, who's a cardiologist and developmental biologist. And we had made with Steve Albelda's lab many years ago a CAR targeting fibroblast activation protein. And John's lab showed that the cells in patients with congestive heart failure and as a repair, they have fibroblasts that express the FAP-CAR target, and they have a cardiac failure model in mice where the mice are made hypertensive and stressed, and over the period of several weeks develop heart failure. Next slide. So what we did then was to treat mice with CAR T cells or control cells. So these are syngeneic T cells that are using a gamma retrovirus to express a CAR against mirroring FAP. And what you can see in this paper published a year or so ago is that the CAR T cells specifically prevent and kill the cells that express the FAP molecule, the activated fibroblasts, and that the cardiac function, as shown further in the paper, is maintained. Next. And shown here is ongoing work with John's lab and with Drew Weissman here at Penn, where we're using CD5 targeted lipid nanoparticles to actually manufacture the CAR T cells in the patient. And so in this case, lipid nanoparticles that have CD5 to target the LNP to the T cells, where then RNA-based translation expresses the FAP CAR transiently in the T cells after this. And we've done it in vitro with the LNPs or in vivo. Click. So in vivo, it's quite surprising. I mean, Drew Weissman's lab can get 20% of the T cells in a mouse to express a CAR, in this case using luciferase, also to express CRI recombinase in vivo in mice. So it's very effective to get transient expression of a CAR in mice with this technology. Next slide. And so then we've used the same stress test in mice, angiotensin, phenylephrine to induce heart failure, and then injected the mice with the lipid nanoparticles a week later, and find very similar effects that we found previously using ex vivo manufactured cells and a gamma retrovirus. We can prevent or ameliorate cardiac toxicity. Next slide, or click. Yeah, so, and this shows the same thing with picoserous red staining, that the fibroblasts are killed with cells, CAR cells that are made in vivo in the mice. Click. So in summary, CARs targeting fibrotic targets can perhaps allow repair of injury and preserve cardiac function. The LNPs appear to be very potent, just very similar to the technology used for Moderna and Pfizer and BioNTech. And we think, that you don't need to kill every last cell in the case of targeting fibrosis, unlike cancer. And then the transient expression of the CAR driven by RNA is likely to limit long-term toxicity of this. And in vivo engineering of CAR cells, it looks really promising, as other labs are also now studying. Next. So there are many possibilities that I think the field will see with engineered cell-based therapies. Next. So, really the main thing I've said, CAR T cells are a living drug. They can persist for many, many years, in fact, a decade in patients. The field has not had, transformation of cells after virally induced gamma retrovirus or retrovirus modification. So T cells are particularly safe if they're autologous and can persist long-term. Next. And, many people were involved in this work, both in my lab and other labs here at the University of Pennsylvania, and collaborators at Stanford and UCLA, as well as children's hospitals. So